Hello everyone, Peter Newman with you. RE Insight this week may be my biggest challenge yet. I'm trying to describe the research by Dr. Danica Goggin from RE. Danica does research into 2,4-D and the other auxinic herbicides or phenoxyherbicides, and she's recently published another paper on this. Now, what you need to do before looking at this video, you need to watch the video above, the one that I recorded a couple of years ago about how 2,4-D works and how we make reference to monkeys. So please have a look at that first so that you can understand where I'm coming from a little bit, and then watch this video. So firstly, I need to go through a few of the mechanisms. What Danica did is she studied 11 different wild radish populations, and she found that basically multiple mechanisms are at play, and there's a range of mechanisms in each population. So there's a lot of different mechanisms, and it could be different combinations of different mechanisms giving 2,4-D resistance. So firstly, that part is very difficult. Now I'll focus on a few different parts of research. First, some research that was done several years ago, and this refers to that previous video that I talked about, the one about the monkeys. So what we have here is we have 2,4-D coming into the plant. It enters through a little transporter on the cell membrane. We've got a cell here called OX1. That's the OX1 transporter. So 2,4-D enters the plant, and if you remember from the video that you've just watched about the monkeys, 2,4-D or auxin is the glue that binds to the transcriptional repressor and also the auxin receptor, and removes the monkey off the back, or removes the repressor off the gene, and then we get a lot of gene expression happening all at once. That causes multiple things to happen in the plant, and that affects plant growth and ultimately the death of the plant. And so some US researchers did some research on the weed kochia, and they found an interference of this pathway here. So an interference of how auxin binds to the transcriptional repressor and causing resistance that way. So that's one of our resistance mechanisms. The other resistance mechanism that I'd like to talk to now is the focus of Danica's latest research, and it involves a whole other pathway of 2,4-D killing plants, which is being understood by researchers now. So what Danica has focused on in this research is some plasma membrane receptors. Now it seems likely, and I use the word likely because the research is still ongoing and we're still trying to understand it, but it seems likely that on the membrane of the plants we have these plasma membrane receptors, and they recognise 2,4-D that is outside of the cell. So 2,4-D is recognised by the plasma membrane receptor. Now it doesn't pump the 2,4-D into the cell. What it does is it just sends a message into the cell to phosphorylate a protein, an enzyme. What does phosphorylation mean? It means adding a phosphate molecule onto an enzyme, and it sort of switches it on. So if you can imagine, we have these proteins, these enzymes sitting in the cells, sort of doing nothing. They're turned off. Once they get the message from the receptor, they are phosphorylated and turned on. Now it gets more complex than that, because this phosphorylated protein causes a cascading chain reaction. It then tells another protein to become phosphorylated, and so on down the chain several times, we don't know how many times, until we get to the final protein that is phosphorylated, and then it does something, we don't know what at the moment, to affect plant growth and kill the plant via 2,4-D, via this mechanism. So it's a new mechanism of 2,4-D killing plants that's being understood by researchers now, and as you can see, there's still a bit of research to do there. Now the focus of Danica's research is up here, the plasma membrane receptors. She found a correlation between the number of these plasma membrane receptors and the level of resistance. So fewer plasma membrane receptors that can recognise 2,4-D, we think they recognise 2,4-D, the fewer of them on the cell membrane, the more resistance, because this whole pathway is not happening without this plasma membrane receptor. So that's a summary of what we know about 2,4-D resistance at the moment. As you can see, all very complex, and research is ongoing. Years ago, Danica started with 2 populations of wild radish and found reduced movement of 2,4-D herbicide in those populations, thought she had found the mechanism. Since then, there's been more research, and she started to focus on other parts of it. She went and studied another 9 populations and found a range of different mechanisms, which has led her to this latest research about plasma membrane receptors. What she has found is a combination of mechanisms in different populations. So we know that there will be different levels of resistance and different resistance mechanisms in a whole heap of different radish populations, and there's probably another group of mechanisms that we don't even understand yet. So that's it for now. I hope that clears things up a little bit. It probably doesn't. It's pretty confusing stuff. It's pretty detailed research. I don't fully understand it. I'm glad that we have researchers like Danica and others that do understand this stuff and can help explain our oldest herbicide, 2,4-D, how it works and how plants resist it.